Cell division is the engine of life. The opening frame makes that point beautifully. All organisms begin as a single cell. Growth and reproduction depend on repeated cycles of duplication and division. And one parental cell can eventually give rise to millions of cells. So the cell cycle is not just a biology topic, it is the logic behind growth itself. The cell cycle has one clear goal: duplicate the genome, synthesize the needed cell constituents, and divide into two identical daughter cells. The wheel diagram shows the major stages: G1, S, G2, and M. And it also reminds you that the cycle is timed differently in different organisms. Human cells divide roughly every 24 hours, while yeast can move through the cycle in about 90 minutes. That timing contrast is worth remembering because examiners love comparing fast and slow cell systems. A huge part of the cell cycle is actually hidden. About 95% of the time is spent in interphase, which is often mistakenly called the resting phase. It is not resting at all. It is the period of intense preparation, growth, and DNA replication. The remaining 5% is the dramatic M phase, the visible division phase. The key lesson is simple. Most of the real work happens before the cell visibly divides. The G1 phase is the growth engine of the cell. This is the interval right after mitosis and right before DNA replication begins. The cell is metabolically highly active and continuously grows. At this stage, DNA has not yet been replicated. That distinction matters because G1 is about preparing the cell to commit to duplication, not about duplication itself. The S phase is the great duplication phase. DNA synthesis takes place here, and the amount of DNA per cell doubles. DNA replication happens strictly inside the nucleus. In animal cells, centrioles also physically duplicate in the cytoplasm. That's a useful dual point. Nuclear DNA replication and cytoplasmic centriole duplication happen together, but in different compartments. The S-phase math is one of the highest yield ideas in the chapter. DNA content changes from 2C to 4C, but chromosome number stays the same at 2N. That is the trap. DNA doubles, chromosomes do not. So if a cell starts as diploid in G1, it is still diploid after S phase. The cell has copied its genetic material, not increased its chromosome count. The G2 phase is the final assembly zone. DNA is already fully replicated, and the cell now synthesizes proteins needed for mitosis. It also completes final cell growth and reaches the critical mass needed for division. So G2 is not a second copy phase. It is the preparation phase for the actual mitotic event. The G0 phase is the quiet exit ramp. Some cells leave G1 and enter an inactive, quiescent stage instead of proceeding into S phase. Adult animal cells like heart cells or cells recovering from injury may stay here rather than dividing constantly. These cells are still metabolically active, but they no longer proliferate. They can be called back into action by the organism if needed. The kingdom divide is an important distinction. In animal cells, mitotic cell division is almost exclusively limited to diploid somatic cells. In plant cells, mitotic division can occur in both haploid and diploid cells. That is why plants can support alternation of generations in their life cycles. The rare exception highlighted is male honeybees, which can divide haploid cells via mitosis. That exception is exactly the kind of detail that turns up in tricky questions. The interphase matrix ties G1, S, and G2 together in one diagnostic chart. DNA content is 2C in G1, becomes 4C in S, and remains 4C in G2. Chromosome number stays 2N throughout interphase. The main activity shifts across phases. G1 is physical growth and high metabolism. S is DNA synthesis and centriole replication. And G2 is protein synthesis plus final preparation for mitosis. This matrix is the cleanest way to track what changes and what does not. M phase is the dramatic division phase, and it has two parallel streams. One stream is karyokinesis or nuclear division, which includes prophase, metaphase, anaphase, and telophase. The other stream is cytokinesis or cytoplasmic division. It is called equational division because the chromosome number in the parent and progeny cells remains exactly the same. The slide also reminds you that cell division is a fluid process, so the boundaries between stages are theoretical rather than perfectly sharp. Prophase begins the unraveling and reorganization. In the earlier state, the DNA is duplicated but still intertwined and not visually distinct. During early prophase, chromosomal material starts condensing. By late prophase, compact chromosomes are formed, each made of two identical chromatids attached at a central centromere. So prophase is really the transition from diffuse chromatin to visible chromosome structure. The mitotic apparatus is assembled in prophase. Asters form around the centrosomes, and spindle fibers extend between the poles. The slide's memory line is very neat. 2 asters plus spindle fibers equals mitotic apparatus. This apparatus is what will later move and separate chromosomes. Without it, the cell cannot divide correctly. A practical NEET example is shown through the onion root tip problem. The key data is that the initial chromosome number is 16 and that number remains 16 after the M phase. What changes is the DNA content. It goes from 2C to 4C in S phase, then returns to 2C after M phase. The chromosome number never changes. Only the DNA quantity fluctuates. That is the cleanest way to understand cell cycle numericals. The final frame is the setup for metaphase. The DNA is fully condensed, the mitotic apparatus is built, and the poles are established. In other words, the cell is fully prepared for the next step, chromosome alignment at the equator. The chapter has reached the point where division can proceed with precision. M phase is where the actual visible division happens. The title itself signals the focus. Metaphase to cytokinesis. By the time a cell reaches this stage, the DNA has already been copied, the chromosomes are condensed, and the machine is ready to separate one cell into two. That is why this phase carries such high exam value. It is the execution stage, not the preparation stage. The journey into M phase begins at the end of prophase. The nuclear membrane disappears, the nucleolus vanishes, and the chromosomes become completely free in the cytoplasm. The slide's diagnostic checklist is the key. Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope are no longer visible as an intact nuclear system. That moment marks the exact start of metaphase-related organization. Metaphase is the equator alignment stage. Chromosomes are now fully condensed and they line up strictly along the metaphase plate. That central alignment is what makes this stage so easy to identify under a microscope. The memory hook on the slide is especially useful. M for metaphase is M for morphology. If chromosome shape and structure are most visible anywhere, it is here. Chromosome attachment is not random. The centromere is the central structure that holds the sister chromatids together. But the spindle fibers do not attach directly to the centromere. They attach to the kinetochore, which is a small disc-shaped structure on the centromere surface. That distinction is a classic exam trap. The spindle connects through kinetochores, never straight to the centromere itself. Anaphase begins when the centromeres split simultaneously. The sister chromatids separate instantly and the terminology changes too. Once separated, they are no longer sister chromatids. They are officially called daughter chromosomes. That naming shift is important because the chromosome identity changes the moment the split occurs. The migration mechanics are very neat. The centromere leads, pointing toward the pole, and the chromosome arms trail behind, creating a V shape as they move. That V shape is not just a picture. It is the visible result of the pulling force of the spindle fibers. The best mental model is a ribbon pulled from the center. The center moves first, and the arms follow. Telophase is the great reversal. Everything prophase did, telophase undoes. The chromosomes cluster at the poles and lose individuality, becoming a discrete mass again. The nuclear envelope reforms around both clusters. Then the returning structures appear. The nucleolus, Golgi complex, and endoplasmic reticulum reform and reappear. In short, the cell is rebuilding both nuclei after separating the genetic material. Cytokinesis is the final divide. Karyokinesis is already complete, so the cell now has to split the cytoplasm itself. The important difference here is mechanical. Animal cells are flexible and can be pinched, while plant cells are trapped inside a rigid cell wall. Because of that, nature uses two different strategies to divide the cytoplasm. The slide also notes that organelles like mitochondria and plastids are distributed between the two future daughter cells during this stage. The cytokinesis diagnostic matrix makes the difference crystal clear. In animal cells, cytokinesis happens by furrow formation in the plasma membrane. The growth direction is centripetal, meaning outside in. In plant cells, cytokinesis happens by cell plate formation. The growth direction is centrifugal, meaning inside out. The precursor structure in plants is the middle lamella/cell plate, while in animals, there is no equivalent precursor structure. The exam keyword alert on the slide is exactly right. Centripetal and centrifugal are high-frequency terms. If cytokinesis fails, the result is a syncytium. In that condition, karyokinesis happens successfully, but the cytoplasm does not divide. The cell then contains many nuclei in one shared cytoplasmic mass. The classic NCERT example is the liquid endosperm in coconut. That is one of the cleanest biological examples of a multinucleate condition caused by division without cytoplasmic separation. The final review compresses the whole M phase into one visual map. Prophase begins condensation, breaks the envelope, and removes organelles from the visible nuclear organization. Metaphase gives perfect equatorial alignment, complete kinetochore attachment, and maximum condensation. Anaphase brings centromere splitting and daughter chromosome migration toward opposite poles in a V shape. Telophase restores the nuclear envelope, returns organelles, and brings chromosomes back to a decondensed identity. That sequence is the full story of mitotic division, and it is the best final map to revise before a 24-hour exam.